Voice is completed now? Correct. Okay, close this door. I will complete it now. You need not to delete any port. You can directly edit now. Hello students, once again to biology classes. Now to read plant growth and development, chapter 13, right? Line by line NCERT. We can complete this one chapter. Now you have already studied the organisms of a flowering plant. Now what is the, where here it is given plant growth and development. Now what is the meaning of growth and development? Growth means an organisms which increases in size, and growth means it is a irreversible increase in size, that is growth. Development means it may be differentiation. Growth and differentiation, growth, and this is called development. Okay, now here what is growth? Like here it is given, have you ever thought about where and how the structures like root, stem, leaves and flowers, fruit and seeds arise, that to an orderly sequence? You are by now aware of term seed, seedling, plantlets. Hello students, once again we are going to read line by line the NCERT chapter 13th in plant physiology. It is plant growth and development. In this chapter we are going to read about the growth, differentiation, dedifferentiation and redifferentiation, then after development, and responsible plant growth regulators. In this case, intrinsic plant regulator, we are going to read this one, right? So what is given here? That is given what is growth. Growth means generally we think about the increase in size, right? Increase in number, increase in length. They are the different modes of growth. Now what is given in NCERT? Aware of the term like seed, seedling, plantlet and mature plant. Look this statement. Seed, first it is a seed, then when it germinates it forms seedlings. Now seedlings, after germination it forms a young plant which is called plantlet, this plantlet, and then it is converted into a mature plant. This is considered as growth and development, right? Now you have also seen that tree continue to increase in height and in girth over a period of time. Over a period of time. That is called growth per unit time, right? The leaves, flowers and fruits of the same tree are not only have a limited dimension but also appears and after then falls periodically and sometimes repeatedly. It continues, right? So why does vegetative phase proceed flowering in a plant? That is another type of growth, flowering growth, right? All plant organs are made up of varieties of tissue. In there any relationship between the structure of a cell, tissue an organ and their function, they perform. Yes, they have interrelationships, right? Can the structure and functions of these be altered? All cells of a plant are descendant of the zygotes. We know starting either it is animal or plant, it is not for only plants. Zygote, it is the first cellular cell from which all the multicellular animals are formed, right? So it is given all cells of a plant are descendant of zygotes, and the question is then why and how they have different structure and functional attributes. That is according to the cell division, and after division differentiation is there, right? Okay, now here one thing is given. To begin with this, it is essential and sufficient to know that the development of a mature plant from a zygote, that is fertilized egg, follow a precise and a highly ordered succession of events. Different succession of events is there. Like that here in previous chapter we read that a zygote further divide and form a embryo. The embryo, all the embryo cells they are considered as tissue, and it is also called as which one? Promeristem, right? Then this tissue is converted into primary meristem. Primary meristem after differentiation forms the maturation plants, that is mature part of the plants. Some mature part of the plant, they again de-differentiation, like cambium, interfascicular or core cambium, right? They again starts that means division and forms meristematic through de-differentiation, and now again these meristematic tissues convert into a which one? Permanent tissue, and this one is called redifferentiation, right? That is here. During the process here, everything goes on very, which one is, look, growth essential, right now. The question, okay, here it is given, everywhere it is given, to begin with thus it is essential and sufficient to know that the development of a mature plant form a zygote. This essential to sufficient to know that the development of a mature plant from a zygote fertilized egg follow a precise and highly ordered succession of events. During this process, complex body organization is formed that produces root, leaf, branch, flower, fruit, seeds, and eventually they died. This is the lifespan or life cycle. The first step in the process of plant growth is seed germination. First step is seed germination. Now the seed germinate, now it converts into seedlings, and when seedlings germinate, that means it forms a young plant which is called plantlet, and after development this plantlet forms the mature plant which is given in earlier, right? Now the seed germinates when it is also favorable conditions for growth exist in the environment. In the absence of favorable conditions, the seed do not germinate and goes into a period of suspended or resting period. Once favorable condition return, the seed resumes the metabolic activities and growth take place. That means for growth, what is favorable conditions are important, right? In this chapter, now we are going to read about these factors which govern and control the development process, right? And these factors are both internal means intrinsic and extrinsic means external. External means what? Favorable conditions like availability of water, that means moisture, oxygen, right? Some other after nutrients after germination it needs light, or another one is temperature. These are external or extrinsic factors. Internal and intrinsic, they may be genetics, they may be intercellular or intracellular. That means they are maybe genetic, or sometimes it is intracellular, like regulator, plant growth regulators are there. Okay. Now what is given? Here some figure is given. In this figure, what is given here? Here it is given soil line, this one is soil line, okay. Now it is a cotyledon, this one is hypocotyle. Now we know this one is hypocotyle, right? Hypocotyle means radical forms the roots, and this one is called hypocotyle, right? And when stem germinate, this side shoot germinates, shoot. So this side is called epicotyle, right? Now hypocotyle, and this one is epicotyle. The hypocotyle means what? It is cotyledons, cotyle form, cotyledon. Hypo means beneath the soil and epi means above the soil. So cotyledons are there, these are cotyledons, they come outside. Epicotyle hook is their form. Now look here, these are cotyledons, epicotyle, this is hypocotyle. Now when you see this one, it is the development in beans. Can you tell me what type of germination is this? We have two types of germination, right? Hypogeal and epigeal. If these cotyledons, they remain inside the soil, it is called hypogeal germination. Hypo means below the soil. Geal come from geo. Geo means geography, that is soil. And epigeal means what? When these cotyles come outside when these cotyledons, they comes outside the soil, they are referred as epigeal mode of it is a bean, so it shows bean, neeps, that means neem, you know neem trees, they shows epigeal, grams, or we have this peas, they shows hypogeal mode of germination. Now this plant is grown, okay. Now look here. Now you understand what is epicotyle and what is epicotyle and hypocotyle? Hypocotyle means what? Which is lower than, look here, which is lower than epicotyledons, and this epicotyle means above the cotyledons. Below the cotyledons, hypocotyle, above the cotyledons, it is epicotyle. Two types of germination, that is epigeal and hypogeal. When cotyledons remains inside the soil, this one is called hypogeal, and when cotyledons comes out of the soil, so they also perform photosynthesis, right? So this one is called epigeal. Now growth is there. Growth is regarded as one of the most fundamental and conspicuous characteristics of living being. Now what is growth? Growth can be defined as the irreversible, permanent increase in size of an organ or its part, or even an individual cell, right? So it is completed by metabolic process, both anabolic and catabolic, that occur in the expenses of energy. Energy means ATP. For example, expansion of a leaf is growth. Expansion of a leaf is growth, okay. Now here one question is given, how would you describe the swelling of piece of food when placed in water? It is called imbibition. This is not a growth, it is imbibition due to the absorption of water. Now plant growth, generally in next passage it is given, plant growth is unique because plant retain the capacity for unlimited growth throughout their life. Unlimited growth occurs at meristematic or in meristematic tissues. So this ability of the plant is due to the presence of meristem on certain location. The meristematic tissue on certain locations means it is present in apical region of root and shoot or branches. It may be in intercalary regions, that means at the axis of leaves, nodes of the leaves. Another one, it may be if it is a dicot root or shoot, so it is present where? In cambium. So the cell of such meristematics have the capacity to divide and self-perpetuate. The product, however, soon loses the capacity. When they lost the capacity of divide, so these cells, they make the plant body. That means they are undergoing in maturation. After maturation, they form the different tissues. This form of growth wherein new cells are always being added. Now what is this? This form of growth when new cells are always being added, right, to the plant body by the activity of meristem, this one is called open form of growth. This one is called open form of growth. Open form of growth. So in plant body, only open form of growth is there. Open form of growth means what? New cells are always being added to the plant body. First thing is here, right now, and unlimited growth is present in plant. Unlimited growth, it is also present in plant body, right? What would happen if the meristem ceases to divide? Does this ever happen? Does this ever happen? This never happens in case of plant only, because plant they have meristematic tissue. If it ceases, that plant, after some time, plant get died. Now here in earlier classes you have studied about the root apical meristem. We read this one, root apical meristem, shoot apical meristem. You know that they are responsible for the primary growth of the plant. Apical meristem is responsible for primary growth of the plant. Now secondary growth of plant, which tissue is responsible? It is cambium. We know already, right? The primary growth of plant are principally contributed to the elongation of the plant along their axis. Elongation means for length. This one is given here. Shoot apical meristem. This one is root apical meristem. They are vascular cambium. They are responsible for secondary growth, right? So primary growth for elongation, secondary growth for thickness, right? For girth. You also know that the dicotyledonous plant and gymnosperm, the lateral meristem, that is vascular cambium, lateral meristem, which is called vascular cambium. This primary growth for elongation, right? And second, in dicot and monocot, the lateral meristem, cork cambium appears in later in life, and these are that cause the increase in the girth of the organs in which they are active, and this one is known as secondary growth of the plant, right? So for primary growth apical meristem is responsible, and for secondary growth cambium and cork cambium is responsible. Okay. Now here growth is measurable. Can we measure the growth in plant also? Yes, we can measure. Now here growth at cellular level. Now first it is given in cellular level. Cellular level is a cell, a single cell, is principally consequence of increase in the amount of protoplasm. Cell increase in size. Since increase in protoplasm is difficult to measure directly, so one generally measures some quantity which is more or less proportional to it. We can measure, right? Growth is therefore measured by a variety of parameters. How many? Many parameters are there, which are, first one is given increase in fresh weight. First one is given here, increase in fresh weight, dry weight, length, area, volume and cell number. How many are there? Fresh weight, dry weight, increase in length, area, volume and cell number, right? Now some examples are given here. You may find amazing to know that one single maize root apical meristem can give rise more than 7,500 new cells in per hour. Second is given, it is the number of cells which is increased in single hour, right? And size in watermelon, size may increase by up to 3,50,000 times. How much size increase, right? 3,50,000 times. It is a wonder, yes. In the former, growth is expressed increase in the cell number, and the latter expressed in increase the size of the cell. Number cell, 7,500 new cells are there. This is a number, and 3,50,000 times it shows the size, right? Now another example also given, pollen tube. Growth of a pollen tube is measured in term of its length, right? And area means what? Increase in the surface area as denotes the growth of dorsiventral leaves. And you know dorsiventral leaves, where they are found? Dorsiventral leaves, they are dicot leaves, right? So what is given in this passage? It is given, we have 5 different parameters to measure the growth. and it may be what is increase in fresh weight or dry weight when we weight. Second is length, area. Length as for example is given in pollen tube. Area example is given in dorsiventral leaves. Volume, volume is we have size, increase in size, that is watermelon. Cell number, it is given in maize 7500 cells, right. Now phase of growth, here a diagram is given. Now look, A, B, C, D, E, F, G. So phase of growth in our previous chapter when we are reading the where morphology of plant, right now flowering plants. In this case we read the apical part of the roots. The period of growth is generally divided into 3 phases, and that side we read this one. We have 3: meristematic zone, elongation zone and maturation zone, right. If you remind this one, then let us understand this by looking the root tips. The constantly dividing cell both at the root apex and the shoot apex represent the meristematic phase of growth, where new cells are continuously divided. This cell of this region, they are rich in protoplasm, no vacuole is there, no intercellular is there, possess a large conspicuous nuclei is there because most active nucleus is there. Their cell wall are primary in nature also, right, thin and cellulosic with evident plasmodesmatal connections. These cells proximal means just next away from the tip, because at the tip in case of root, which what is there, we have root cap. After root cap we have that is a permanent tissues, cluster of cells, permanent tissue, and just after beneath this one we have this meristematic zone, right. Meristematic zone represents the phase of elongation, right. The meristematic zone, and after this meristematic zone we have phase of elongation and increased vacuolation, where in case of in a phase of elongation the cells which are present in the region of elongation, these cells increased vacuolation, cell enlargement and new cell wall depositions are characteristics of the cell in this phase, right. Phase of elongation means what? New cells are added. Second, cell elongation is there. Vacuolation is also there. Further away from the apex, that is more proximal to the phase of elongation, lies the portion of axis which is undergoing the phase of maturation. I think you remember this one, this structure, previous structure, right, where we have a root. This is root cap, this side meristematic zone, this is elongation zone, and from this zone we have root hairs, right. So this is zone of maturation, this is elongation zone, okay, and this one is our meristematic zone, this one is root cap, right. So consider this one is, it is given here. Proximal means what? Look, this is distal end, right, and this side proximal, right. The proximal to root cap we have meristematic zone, proximal to meristematic zone we have elongation zone, and proximal to elongation we have zone of differentiation or maturation zone, okay. Now another point is there. Look what is given here. Lies the here more proximal to the phase of elongation lies the portion of axis which is undergoing the phase of maturation. The cell of this zone attain their maximal size in term of wall thickening and protoplasmic modifications. Most of the tissues and cell types you have studied, it represents the phase. Now growth rates. So growth rate, we have 2 types of mathematical growth rates. One is called arithmetic and another one is called geometric. So the increased growth per unit time. What is growth rate? What do you mean by growth rate? Growth rate means when we consider the rate, that means we have some times. The increased growth per time unit is called growth rate. Increased growth per unit time, either it is 1 second, 1 minute, 1 hour or 1 day or 1 year. Thus the rate of growth can be expressed mathematically per unit time. an organism that means can produce more cells in a variety of ways. Now a figure is given here. Look the figure. It is arithmetic. What is in arithmetic? Here this side, this one, it is divide. This one is called parent cell, and this side they are another cell. So one cell it continue divided, that means it is meristematic cell, and this one is permanent cell. So one cell, this is germ layer, and this side cell continue growth. So one cell continues to growth, but another it is stopped. Here on both sides, this is also continue growth. This one is look continue to growth, continue to growth. On both sides cells grow. So this one is called arithmetic and this one is called geometric, right? So here what happens in very first time, these have two cells, they have two cells, both have, this is called lag phase. Lag means slow phase, also lag phase, slow phase. But after then enormous growth is there, very rapid growth is there. So rapid growth means what? Rapid growth means log phase. Lag phase, log phase. But after some time what happens? If we have limited resources, then it also slow down and ultimately it forms S shape. Sigmoid curve is there, and here a linear curve. Here linear structure is formed, but in this case a S shape, that means this shape is formed, and this one is called sigmoid curve. Okay, so this is the diagrammatic representation here, right? One figure C is given here. C, it represents the zygote division. Look here, zygote divide into two cells, 1 and 2 cell. Now these cells are divided, both cells are divided, and further they form the 8 same. So this is a geometric phase, means all the cells are divided, right? But after then some cells are dividing, but not all cells. Some cell they undergoes into maturation, right? And some cell they continue to divide. So cells capable of division and cells that lost the capacity of division, that means capable of division, they forms meristematic region, and these are permanent region, right? Permanent region. So what happens here? Here arithmetic phase of development is there, arithmetic phase of growth is there. So both type of growth is there. This side geometric, and after some time arithmetic is given, right? Arithmetic growth is there, and it is also given in this diagram, right? Diagrammatic representation of arithmetic geometric growth. This one is arithmetic, this one is geometric, and this is both stage during embryonic development, showing geometric, initially geometric, and further it convert into arithmetic phase. Now here it is given, the growth rate shows an increase that may be arithmetic or geometrical, as we see in the first. Arithmetic means one-sided development, geometric means both-sided development, growth is there. The arithmetic growth following mitotic cell division only, look here, following mitotic cell division, right? Following mitotic cell division, only one daughter cell continues to divide while the other differentiate and matures. And the simplest expression of arithmetic growth is exemplified by a root elongation at a constant rate. Root elongation. Only one cell continue to divide, another cell it goes into maturation. First elongation, then maturation. Right? On plotting the length, this one in a graph, mathematically it is expressed by LT is equal to L0 RT, in which LT is the length at the time t, after the time t, L0 length at the time 0 means initial time, R means growth rate per unit time, right? This time height of the plant is here, and this is time, either it is in hour or second, this is the height of the plant increase. So we found this a linear, it is called linear graph, right? A straight line is there. Let us know what happens in geometrical growth. Now second one we have geometrical growth, right? In geometrical, what happens? The initial growth is slow, lag phase, because only small number of cells are there, and it increases rapidly thereafter at an exponential rate. Exponential means rapid growth is there, log or exponential phase it is called. Hence, here both the progeny cells following mitotic division, what is given? Both the progeny cells. In arithmetic only one cell, but here both cells, right? They continue the divide, retain the ability to divide and continue to do so, however with limited nutrient supply. When nutrient supply is limited, when it is unlimited, they form J-shaped, right? When it is what is given here? Look, when it is limited, so when limited, this S shape, sigmoid shape, this is called sigmoid curve is formed when it is limited, but when it is unlimited, we got this J shaped, right? So this one is called J shaped curve. When it is unlimited, it is for unlimited, and it is for, this one is for limited, right? So because unlimited is not present, it is not present in our that is habitat, because each habitat has a limited resources. So this sigmoid, this one is real growth. If we plot a parameter of growth against time, so we get a typical sigmoid or S curve. A sigmoid curve is a characteristics of living organisms growing in a natural environment. It is a typical for all cells, tissues and organs of the plant, right? This is a natural. Now it is given, can you think of more similar examples? So more similar examples, seasonal growth. In winter season, plant growth is slow, but in summer, plant growth is very rapid. So slow, rapid, slow, rapid, slow, rapid, this forms sigmoid curve. It is given here. What kind of a curve can you expect in a tree showing seasonal activity? It is sigmoid or S-shaped curve. The exponential growth can be expressed as W1 is equal to W0 RT, in which W1 means final stage, final size, either weight, height, number, extra, it is depend upon our consideration. Now W0 means initial size at the beginning of the period, R for growth rate, T for time of growth. Growth rate means in per time area, right? And E is the base of natural logarithms, right? Now here R is the relative growth rate and is also measure of the ability of the plant to produce new plant materials, referred to as the efficiency index. Where hence the final size, so this you understand this one, it is for exponential phase. Here lag phase is there, look. This one is called lag phase, this time it is log phase, right? And this one upon it is stationary phase. Right? Lag phase, log phase, stationary phase, when it becomes constant, when the growth become constant. Now here R is the relative growth rate and it is also measured of the ability of the plant to produce new plant material. Now here hence the final size of referred to as efficiency index. One thing is given, R is the relative R is the relative growth rate and is also the measure of ability to the plant to produce new plant material, which is referred as efficiency index. So R is also considered as efficiency index. Hence the final size of W1 depend on the initial size W0. Now W1 is totally depend upon W0, initial size of the, right? Now here an example is given, also a diagram is given here. We have two leaves. Look at these leaves. Now here it is given two leaves and both it is 5 cent, initially it is 5 centimeter, 5 centimeter, and after some time, after unit time, it becomes 10 centimeter. Now when we consider what is the absolute growth, the absolute growth is 10 minus 5, it is 5 centimeter, right, square. It is absolute growth. Here also we have what is final 55 centimeter square, and first it is initially it is 50 centimeter square. Now it has also how much 5 centimeter absolute growth. Both have equal absolute growth, right? But one another thing is, and relative growth rates. Now growth, what is the growth rate? When we consider this growth rate, that means per time. So now what is the percentage of growth? Here 5 centimeter is given. Now initial we have 5 centimeter. So 5 upon 5 multiply by 100. Now we have how much? 100% growth is there. 100% growth is there. This one is relative growth rate, right? But when we consider this one, we have how much? It is 50. So I write here 5 centimeter. This one is the absolute growth, right? Initially we have 50 centimeter. Here initially is 5 centimeter. Multiply by 100. Now we obtained it is 2 and 10% relative growth is there. So that is the difference between absolute growth and relative growth, right? Absolute growth of both leaves are equal, but relative growth means it is different. In A, it is 100% relative growth is there, and in case of B, only 10% relative growth is there, right? Okay. Now here quantitative comparison between the growth of living system can also be made in two ways. First, measurement and the comparison of total growth per unit time is called absolute growth. As we here 5 equal, both leaves have equal, that is 5 centimeter only, and the growth is the given system per unit time express the common basis per unit initial parameter is called the relative growth rate, and this is 100% and 10%, right? Figure is given here. Two leaves A and B are drawn that are different size but shows absolute increase in area in the given time to give leaves A and B1. However, one of them shows much higher relative growth. Which one and why? Now here we come to know that which one is A. This is given A. So A, that is small leaf, it shows us more 100% and B shows only 10%. Which one is more much higher relative growth? A, right? And why? Because when we consider the interrelation between the primary leaf and the after maturation, that is final leaf, so we find out this ratio, this percentage. Now condition for growth. What are the essential condition for growth? Now essential current condition we have here mainly 3 conditions is there: water, oxygen and nutrients. So this list may be are as very essential element for growth, right? Plant cell grow in size by cell enlargement, which is turned in requires water, right? Turgidity of cell helps in extension of growth, otherwise leaves get curling. Thus, plant growth and further development is intimately linked to the water status of the plant. Water also provides the medium for enzymatic activities, what is very essential, right, for the growth. And oxygen help in releasing metabolic energy, that is Krebs cycle aerobic respiration for energy, right? Energy essential for growth activities. Nutrients, they are macro and micro essential elements, like we know very well, they are 17 in numbers, right, are also required by plant for the synthesis of protoplasm and act as source of energy. So 3 things are essential. Which one? Water, oxygen and nutrition. Another, in addition, every plant organism has an optimum temperature. This one temperature. So it need temperature, optimum temperature range, right, to best suited for its growth, because we know here our plants grow 10 times more rapidly than we consider if tundra plants, Siberian plants, right? Why? Because here optimum temperature is there. Second, environmental signals such as light and gravity also affect the phase or stage of growth. Now here we are given water, oxygen, nutrients, optimum temperature, light and gravity, right? They are responsible or they are essential for growth. Now here differentiation, de-differentiation and re-differentiation. What is differentiation, what is de-differentiation and what is re-differentiation? So here I am going to tell you about from this example. Suppose that here we have what is, we have primary meristem, right? This primary meristem further forms mature permanent tissue. So this step is called, this is called a differentiation. Differentiation, right? The maturation is the result of differentiation, right? Then after this mature cells, some of the mature cells, they convert into once more secondary meristematic tissue. Meristematic or secondary meristem. Now this time it is called de-differentiation. De-differentiation. Here it is called de-differentiation, right? TATION. Now secondary meristem, after maturation it forms once more permanent tissue, right? And this time it is called re-differentiation. As for example, primary meristem forms permanent tissue. Here mature permanent tissue, they are parenchyma. Parenchyma. In case of where in medullary rays, that is called in the root, right? Na? Medullary rays is there. These cells are parenchyma cells which are permanent tissue. Now these parenchyma cells in dicots, they are converted into interfascicular cambium. So interfascicular cambium, that is parenchyma cells which are already permanent cell, after maturation through differentiation, they once more convert into meristematic tissues, or in cambium, they convert into cambium, right? Now this cambium, it is called interfascicular cambium. Interfascicular cambium and cork cambium, because they once they are permanent tissue in the form of parenchyma cells, they convert into meristematic cells, cambium cells and cork cambium cells. It is called de-differentiation, right? And when this meristematic cells, that is interfascicular cambium or cork cambium, once more they convert into phloem or xylem, that is permanent tissue, this one is called re-differentiation. It is given here also, right? The cell derived from root apical and shoot apical meristem and cambium differentiate and matures to perform specific function. We know this one. This act leading to maturation is termed differentiation. What is differentiation? This act leading to maturation, the act which lead to maturation, right? Act which leads to maturation is termed as differentiation. Look here, differentiation, a mature permanent tissue is formed. During differentiation, cell undergo few major structural changes both in their cell wall and protoplasm. For example, to form a tertiary element. Tertiary element means trachea. Trachea means what? We know very well. These are xylem vessels, right? The cells would lose their protoplasm. They also develop a very strong elastic lignocellulosic secondary cell wall to carry water to long distance under extreme contentions. So try correlated the various unknown. Okay, here now plant shows another. This one is differentiation. Maturation is termed as differentiation, right? Now plant shows another interesting phenomenon. The living differentiated cells that by now have lost the capacity to divide can regain the capacity of division under certain condition, and this phenomenon is termed as de-differentiation. Look here, I write here, these mature permanent tissue, they convert into secondary meristematic tissues, and this one is called de-differentiation, right? Meristematic tissue convert into permanent tissue or mature into permanent tissue. In the process of maturation, it is called differentiation. And when the permanent tissue convert into once more meristematic tissue, this one is called de-differentiation, right? And when this secondary meristematic tissue convert into once more permanent tissue, this one is called re-differentiation, right? So this is sequence: first differentiation, de-differentiation, and then re-differentiation. For example, formation of meristemate interfascicular cambium and core cambium. Form fully differentiated parenchyma cell from fully differentiated parenchyma cell. It is given here. Now, while doing so, such meristem tissue are able to divide and produce cells once again lost the capacity to divide, and produce cells that once again, okay, but mature specific function. What is given here? They once again, look here, this meristematic tissue, they once again convert into permanent tissue, and this is called redifferentiation, right? This once again lost the capacity to divide but mature to perform specific function, that is called redifferentiation. As for example, secondary phloem, secondary xylem, they are the example of redifferentiation. List of the some tissue in the woody dicotyledonous plant. The wood is the product of redifferentiation. Bark is the product of redifferentiation, right? Now here given, how would you describe a tumor? So tumor is also a, what is tumor? How a tumor is formed? Tumor means what? First it is mature cells again regain, right now again starts division, that means they regain the capacity of division, that is called tumor. So this is called de-differentiation. What you call the parenchyma cells that are made to divide under controlled laboratory condition during the plant tissue culture, so we set them these, so we set them de-differentiation. It is called de-differentiation, right? Because they are divide, and when they convert into that is mature plant or permanent tissue, then we set them redifferentiation, right? Okay, recall in section 13.111, okay, we will see this one. We have mentioned that the growth in plants is open. Open means what? It can be indeterminate or determinate. It is given here. Look, growth in plant is open and it can be determinate or indeterminate, right? Open means what? Continuous new cells are added. Now determinate and indeterminate means what? Indeterminate means we cannot determine how much time that means it is not determined, right? Now we may say that even differentiation in plant is open. Even differentiation in plant is open because cells or tissue arising out of the same meristem have different structure at maturity. The final structure at maturity of a cell or tissue is also determined by the location of the cell within it. What is the location of cell? We can determine this one, but right, for example, cell positioned away from the root apical meristem differentiate as root cap cell. That means if it is a calyptrogen, in case of monocot, it forms the root cap cell. We determine this forms the root cap cell, right? In case of where monocots. In case of dicot, dermatogen is there, so they form the root cap. While those pushed to the periphery mature as epidermis. Can you add a few more examples of open differentiation correlating the position of a cell to its position in an organ? Yes, we can, right? One example is like tunica carpus. In case of this is there, it is given, this one is given for roots, and when we consider the shoot, so we have tunica carpus. So tunica, the cell of outermost, that is location, outermost cells, they forms tunica. Tunica means what? Epidermis. So they, after some time, they are converted into epidermis, right? Now development. What is development? So development means what? Growth and differentiation. What is given? Development is a term that includes all changes that an organism go through during its life cycle from germination of the seed to senescence, that means olden age. Diagrammatic representation is given here. What is given here? Look this one. Meristematic cell, zone of cell division, right? Now plasmatic growth means expansion or elongation of cell, this one. Meristematic cell, cell divide here. Now cell after division, elongation zone is there, it is called plasmatic growth. This one expansion or elongation, right? Then maturation zone is there. Maturation means they are differentiated into mature organs or tissues. Now mature cells, after some time, they become old, it is called senescence, right? And sometimes it is the death, right? So this one is cell division. Sometime what happens? This elongation, maturation, they starts de-differentiation and start division. They once again start division. So once again start division, that means they becomes meristematic cell. And then plasmic, same process occurs. Expansion or elongation is there, plasmatic growth, and then after re-differentiation occurs. And after re-differentiation, they get mature, and then old age is there. Now we understand this one. Look here. This one is meristematic zone. Maybe primary meristem. In primary meristem, plasmatic growth is there, expansion, same process is there, right? Okay. Now here they are not, they convert into differentiation. After differentiation, they convert into maturation, and these mature cells after some time they become old and died. Okay. Another function is there. Meristematic zone. This is primary meristem. They after expansion, elongation is there. Then after maturation, they mature. In differentiation is there, right now. Some cell of mature, like in parenchyma cells, in case of medullary rays, in case of dicot plants, they differentiated into which meristematic zone? That is secondary meristem. Now secondary meristem is there, its cell division is occurs, right? Now it becomes secondary meristem. Once more plasmatic growth, expansion is there, then redifferentiation is there. Now it becomes redifferentiation. Here dedifferentiation. After redifferentiation, they form mature cells, and these cells after some time, after old, become old and then dead. So either for dedifferentiation or from that means promeristem, meristematic cells are formed. Okay, first question is there from myself. What do you mean by meristematic cells? We have two types. I said you already, promeristem, primary meristem and secondary meristem. So promeristem is embryonic stage, right? After the growth of plant, we have primary meristem, and primary meristem is responsible for the formation of primary organs, right? So primary meristem here, meristematic cells is given, it is not primary or secondary, it may be primary or secondary, same type of graph is there. Primary meristem is there. Now plasmatic growth, after differentiation, mature tissue is formed, and after some time it becomes old and further died, right? Okay. Some cells of primary meristem, after differentiation, they mature and form a tissue like parenchyma tissue. Through dedifferentiation, they once more forms meristematic cells, that is called secondary meristem. And now secondary meristem, if secondary meristem is there, their cell division occurs, elongation is there, and after then redifferentiation occurs. Here redifferentiation and maturation, mature cells form, become old and dead. So for both, this one is correct, right? Now here given plasticity. What is plasticity? Plant follow different pathway in response to environment or phase of life to form different kind of structures. This ability is called plasticity. A same part of plant, it shows to different type of structures in different habitat, in different phase of life. So this one is called plasticity. Plasticity. This ability. What is the ability? Plant follow the different pathway in response to environment or phases of life to form different kind of structure, and this ability is called plasticity. As for example, heterophily. Hetero means different, phily means leaf. Different leaf in case of cotton, coriander and larkspur. It is given example, right? In such plant, leaves of juvenile plant, they are different shape from those in mature plants. Another example is also given for habitat or climatic. On other hand, difference in shape of leaves produced in air and those produced in water in case of buttercup also represents the heterophily, different type of leaves, development due to the environment. This is called plasticity. Examples is given also here and figure is also given. Look, this one is the coriander leaves, juvenile coriander leaves, and this one is adult coriander leaves in different phases, development phase. And this one is our buttercup right now, like look here, terrestrial, and this one is water habitat, different. So this is the example of plasticity. Thus growth. Now what is growth? Differentiation and development are very closely. Growth, differentiation and development are very closely related event in the life of a plant. Broadly, we can say development is considered as the sum of growth and differentiation. What is development? That means what? Development is considered as the sum of growth plus some differentiation, right? So development in plant, both growth and differentiation is under control of intrinsic as well as extrinsic factor. Intrinsic means internal, extrinsic means external factors. The former include both intracellular, that is inside the cell, intracellular means inside the cell, that is called genetics as for example, or intercellular, that means outside the cell factors. They are chemicals such as plant growth regulators, or we said them plant hormones, right? While the latter includes light, latter means what? Extrinsic. Extrinsic include light, temperature, water, oxygen and nutrients. These are external, right? So what is given here? Here it is given, they may be intrinsic. Intrinsic means we have two types of intrinsic, either intracellular, INTRA, inside the cellular, or intercellular, outside the cell. So these are plant growth regulators and this is genetic, right? And another one is extrinsic. So extrinsic means what? Extrinsic means we have 5 things are given here: light, temperature, water, oxygen, right, and nutrition. Now plant growth regulators, characteristics. The plant growth regulator, that is commonly called as PGRs, are small simple molecules. They are small simple molecules of diverse chemical composition. They could be indole compounds like indole acetic acid, indole 3-acetic acid, right? Indole 3-acetic acid, commonly IAA or auxin, we said them, or adenine derivatives and 6-furfurylamine purines. Look here, adenine derivatives, you know adenine, adenine means what? It is a nitrogen base, right? And furfurylamino-purine, commonly called as kinetins like cytokinin. Derivatives of carotenoids, as for example abscisic acid, ABA, terpenes, gibberellic acid, gibberellic acids, GA3, it is the first observed growth regulator, GA3, or it is in the form of gaseous ethylene C2H4. Plant growth regulators are variously described as plant growth substances or plant hormones or phytohormones in literature. Look here, these PGR, they are also described as plant growth substances, plant hormones or phytohormones. Phyto means already plant, right? Now the PGRs can be broadly divided into two groups based on their function on a living body. So these two groups may be growth regulator or growth inhibitor. One group involving growth promoting activities, that is called growth regulator, such as cell division, cell enlargement, pattern of formation, tropic growth, flowering, fruiting or seed formation. So they are considered as growth promoters, plant growth promoters. As for example, we have auxin and gibberellin and cytokinins. Right? Auxin, gibberellin and cytokinins. Another group important role in plant responses to wound. It is a stress to wounds and stresses of biotic and abiotic origin, and they are also involved in various growth inhibitors activities such as dormancy and abscession. These PGR abscisic acid belong to this group. But for ethylene which is a gas could fit either of the group. Either we can consider as plant growth regulator or we can consider as plant inhibitors. But one thing is there, this ethylene, could fit either of the group, but largely it is an inhibitor of growth activities. So when we have only two group, so one auxin, cytokinin and gibberellic acids, they belongs to growth regulators, and abscisic acid and this one ethylene, they belongs to growth inhibitor. But in case of ethylene, what happens? It can be, that means related with growth regulator also, but most of the function it is inhibitory functions. Okay. Now the discovery of plant growth regulators, how they discover? It is accidentally. All the plant growth regulators, they are discovered accidentally. Interestingly, the discovery of each of the 5 major groups of PGRs have been accidental, and this started with the first observation is Charles Darwin. You know very well this one, you are familiar with this name, Charles Darwin, and his son also. His son's name is Francis Darwin. So when they observed that the coleoptiles of canary grasses, you know very well coleoptiles. When we read the seed, so we have coleoptile and coleorhiza. The shoot on the shoot side we have coleoptile, and the root side it is coleorhiza, right? So this coleoptiles of canary grasses means it is monocots, responded to the unilateral illumination by growing toward the light source, that is phototropism. They shows the phototropism. After a series of experiments, it was concluded that the tip of coleoptile was the site of transmittable influence that caused the bending of the entire coleoptile. Which one tip? That means tip has some specific chemicals or some substances which are responsible for phototropism, and these substances, they are crystallized, and after some time it is identified that they are auxins. And first auxin was isolated by F.W. Wendt from tips of coleoptile of oat seedling. F.W. Wendt. So for auxin, in the discovery of auxin, we have 3 scientists. First Francis Darwin, first Charles Darwin, father, and his son, it is Francis Darwin, and it is isolated by F.W. Wendt, which is isolated what auxin is isolated by. Second, Bakane disease. Bakane, it is called foolish seedling of rice seedlings, was caused by a fungal pathogen. This fungal pathogen, it is called Gibberella fusiculae. Gibberella fusiculae, and scientist named E. Kurosawa, 1926, he reported that the appearance of symptoms of the disease in the rice seedling when they were treated with sterile filtrates of the fungus, the active substances. Now, right now, so this rice seedling, this disease, when the extract of this fungus is sprayed on this rice, so they shows the Bakane disease, right? And this active substance was later identified as gibberellic acids, Ga3, because name come from Gibberella fusicori. Now F. Cook and his co-worker, third form is there, observed that from the internodal segment of tobacco stem, the callus means what? A mass of undifferentiated cell. Callus means what? A mass of undifferentiated cell, right? Proliferated if in addition to oxygen, the nutrients medium was supplemented with one of the following. Which one? Extract of vascular tissues. Second one is yeast extract or coconut milk or DNA. When we provide these supplement, right, so their callus, it is proliferated. Miller et al. In 1955 later identify and crystallize the cytokinesis promoting active substances, and they termed the kinetins, right, during mid 1960. Now after this Miller et al. In 1955 and after 5 years in 1960, 3 independent research reported that purification and chemical characterization of 3 different kind of inhibitors, that is inhibitor B, this one, inhibitor B, abscission second or dormin. In different three different scientists in 3 different region of the world, they that means purify these 3 chemicals, and later all the 3 were proved to be chemically identical and name is given abscisic acid, right. So initially they are different names, inhibitor B, abscission second and dormin, and finally only one name is given, it is abscisic acid. And who is responsible here? Abscisic acid, this is the plant inhibitor, right. Now H. Cousins 1910 confirmed the release of volatile substance, that means gaseous form, from ripened orange that hastened means rapid the ripening of stored unripened bananas. Later this volatile substance were identified as ethylene, a gaseous PGR, right. So this is the discovery of different PGRs, oxin, then after E. Kurosawa, it is gibberellin, then after who? Skoog, this is kinetins, then abscisic acid, and last gaseous PGR. Who discovered this gaseous PGR? H. Cousins. Okay. Now what is the effect of these plant growth regulator on plant? First thing is given, physiological effect of plant growth regulator. First we have auxin. Auxin means indole 3 acetic acid or its derivatives. So we have natural and artificial, both type of auxins are there. Auxins from Greek auxin, to grow, was first isolated from human urine. This one is very important statement which is asked in various time. From auxin was first isolated from human urine, not from plant but from human urine, right. The term auxin is applied to the mindole 3 acetic acid IAA, it is the natural. Look, we have 2, IBA and IAA, both are natural, and all the other they are artificial, like. And to other natural and synthetic compounds having certain growth regulatory properties, they are generally produced by the growing apex of the stem root, that means in apical region, meristematic tissue, from where they migrate to the region of their action. Auxin, which one? Like IAA, indole 3 acetic acid, and indole butyric acid IBA have been isolated from plants because they are natural, so they are formed inside the plant, right. NAA, naphthalene acetic acid, and 2,4-D. 2,4-D means what? 2,4-dichlorophenoxy acetic acid, right now. So this 2,4-D, it is a what is called BD side for dicots only, are synthetic auxin, right. So here given example is given here, 2 examples for natural, that is IAA and IBA, and 2 for synthetic, that is NAA and 2,4-D. All these oxygen have been used extensively in agriculture and horticulture practices, right. Agriculture you know very well, horticulture means fruit and vegetable and flower, cultivating. They help in initiate rooting system in cuttings. First is given here. What is the function of this auxin? Auxin is, in case of tissue culture, it is used. Tissue culture and also in vegetative propagation. They help to initiate rooting in stem cutting, that is stem propagation. An application widely used for plant propagation, vegetative propagation in case of stem cutting, like in rose or in sugarcane. Auxin promote flowering in pineapples, help to prevent fruit and leaf drop at early stage. But if leaf and fruit are old, then it promote abscission of the older mature leaves and fruits. You understand what are the functions given here? Here given, in case of vegetative propagation, auxin induced roots, right? It also promote flowering in case of pineapple. Third is given here, first young leaf and fruit, it prevent to drop, but if they are old, so abscission, it induce the abscission. Abscission means to separation, right? Fall of the leaves and fruits. In most higher plants, the growing apical bud inhibits the growth of lateral axillary bud, and this phenomena is called apical dominance. Look here, the growing apical bud inhibits the growth of the lateral or auxiliary buds. This phenomenon is called apical dominance, right? Because they suppress the growth of lateral branches. Removal of shoot tip, note is given. It is called decapitation. Capitation, cap means roots. This is two tips. When we cut these shoot tips, usually results in the growth of lateral buds, and it is widely applied in tea plantation, hedge making, right? Because when we cut this tip, now lateral, this apical dominance is absent. Now lateral branches grow. Auxin also induced parthenocarpy. What do you mean by parthenocarpy? Parthenocarpy means seedless fruit. Without fertilization, fruit is formed, right? So auxin also induced parthenocarpy in making seedless fruit, as in tomatoes, grapes also, papayas also, right? They are widely used as herbicides. I already tell you, 2,4-D is usually used as herbicides and on dicot plants. Example is given here. They are widely used as herbicides. 2,4-D widely used to kill dicotyledon weeds, does not affect the mature monocotyledon plants. It is used to prepare weed-free lawns by gardeners, because in lawn they are made up of grasses. Grasses means they are only monocotyledon plants, right? Auxin also control xylem differentiation and help in cell division. Xylem differentiation, who is responsible? Auxin, and also helps in cell division, right? So these are the function of auxin. Now we have gibberellins. Gibberellins. So what do you mean by gibberellins? Gibberellins here, another kind of promotory PGR. They are more than, this time we have more than 100 gibberellins reported from widely different organisms, mostly like fungi and higher plants. They are denoted as GA1, GA2, GA3 and so on, right? But we cannot consider, this one is there, we cannot consider GA1 is the first gibberellin which we discovered. First gibberellin is GA3 which was discovered, right? Not GA1. So they are denoted as GA1, GA2, GA3 and so on. However, gibberellic acid GA3 was one of the first gibberellin to be discovered. Now you understand this one, GA3, and remains the most intensively studied from all GAs are acidic. That's why we are said them gibberellic acid, right? So all these are acidic. One thing is there. They produce a wide range of physiological responses in plant. Their ability to cause an increase in length of, because they are responsible for elongation, mostly elongation, right? So they are cause an increase in length of axis to use to increase length of grapes vine, right? Grapes stalk. Gibberellin cause fruit like apple to elongate and improve its shape and size also, right? Gibberellin, another one is given, they also delay senescence means old. Thus the fruit can be left on the tree longer so as to extend the market period. Longer means it is available after the natural period. GA3 is used to speed up the malting process in brewing industry, breweries, formation of alcohols, right? Wine industries. Sugarcane, now sugarcane we know very well for improving the quantity per acre. We spray this gibberellin and the internode become elongated. When internode elongated means more sugarcane plant is there. More sugarcane plant means more yield is there. So sugarcane store carbohydrate as sugar in the steps. Spraying of gibberellin on sugarcane crop increase the length of the stem and thus it increase the yield by as much as 20 tons per acre. Right? Spraying juvenile conifers with GA, it hastened means rapid the maturity period and thus leading to early seed production. Right? Also it promotes the bolting. Bolting effect means what? Very fast. Right? Node and internode. Internode is growing very fast. It is called bolting effect. Also promotes bolting, internode elongation just prior to flowering in beet, cabbage and many plant with rosette habit. Cabbage, you know, rosette habit is there, very small internodes are there, and this. Okay, now the another plant regulator is cytokinins. The cytokinins have a specific effect on cytokinesis. Cytokinesis means cell division. This is for cell division. As we are discovered as kinetins. How much? I have one heading, this one ethylene, and then finish. Can we? Storage. Okay, only this. Then this is finish. Can we continue? Otherwise, and only one heading is there. I need 10 minutes. Huh? So after 10 minutes, this chapter is completed. Otherwise, we have to make one and more video. I want to complete this one. No? Okay. So this time you will cut this one. Only one. And remaining no cutting there, right? Now cytokinase. Cytokinase have a specific effect. Okay, cytokinesis, as I told you, cytokinesis is what? Cell division. So cytokinin is responsible mainly cell division. Gibberellin, it is responsible mainly elongation, right? And auxin mainly responsible for differentiation, right? Here, they were discovered as kinetins, a modified form of adenine A purine, and 6-furfurylamine purine, right? From the autoclaved herring sperm DNA, kinetin does not occur naturally in plants. In another form, they are formed. Search for the natural substances with cytokinin-like activities led to the isolation of G18. It is called G18. G18 means a protein. GEA, it comes from G maize because it is first observed in maize. G18, several corn, okay, G18 from corn kernels and coconut milk. Since the discovery of G18, several natural occurring cytokinins and some synthetic compounds with cell division promoting activities have been identified. Natural cytokinins are synthesized in the region where it is synthesized because they are not a specific structure is there. They are in different plant. They have different structures. So we said them kinetins, right? Where rapid cell division occurs, they are present there because they are only for responsible for cell division, right? So for example, root apex, shoot apex, developing shoot buds, young fruits, here cell division occurs. So they are present in that region. It also helps in produce new leaves, chloroplast in leaves, lateral shoot growth, and adventitious shoot formation, not root, shoot formation. Adventitious shoot formation. Cytokinin help overcome to apical dominance. That means they are antagonistic. This cytokinin it is antagonistic to auxin, right? They promote nutrient metabolism which help in the delay of leaf cells. Okay. Now another we have ethylene. This is a gaseous PGR. Ethylene is a simple gaseous PGR. It is synthesized in large amount by tissue undergoing senescence and ripening fruits. Senescence means which are undergoing old age, right? and ripening fruits. Influences of ethylene on plant include horizontal growth of seedling. Now it is given the what is the functions of this one. Influences of ethylene on plant include horizontal growth of seedling, swelling of the axis and apical hook formation in dicot seedlings after growth, right? Ethylene promotes also senescence and abscission of plant organs, especially of leaf and flowers. For ethylene is highly effective in fruit ripening, so we use ethanols right now. In ethophanes, right? It enhances the respiration rate during ripening of the fruits, and this rise in rate of respiration is called respiratory climactic. Increase in respiratory rate due to the presence of this ethylene in case of fruit. Now fruit get ripened. Ethylene breaks seed and bud dormancy. It breaks. Initiate germination in peanut seed, sprouting of potato tubers. Peanut and sprouting of potato tubers. Ethylene also promote rapid internode or petiole elongation deep water rice plant. Rice, we know very well, rice grow in deep water. When we increase the level of water, rice plant also increases. So what part of rice plant increases? Internode, and this one is given petiole. Petiole means it is the leaf stalk, right? They elongated, that means it, and this helps the leaf and upper part of the shoot to remain above the water. They can survive. Ethylene also promotes root growth and root hair formation, thus helping in the plant to increase their absorption surface. So this side, in this passage, we it presents the growth characteristics of ethylene, and above passage it shows the inhibitor properties, right? Ethylene is used to initiate flowering for synchronized food set in pineapples. It also induces flowering in mango, right? In pineapples, induce flowering in mango. Since ethylene regulates so many physiological processes, it is one of the most widely used PGR in agriculture, in horticulture, agriculture also. A most widely used compound of source is ethylene is ethone, right now. Ethone is an aqueous solution, is readily absorbed and transported within the plant and release ethylene slowly. Ethophane hasten the fruit ripening in tomatoes, apples, accelerate abscission in flower and fruits, that is thinning of cotton, cherry, walnut, and also promotes female flower in cucumbers and therefore increase the yields, because we know cucumber means we have unisexual flowers, male and female. It increase the number of female flowers. The female flowers, they develop into fruit. Cucumber means or the gourds, right? Gourds, they may be gourd, pumpkin, beetle gourd, ridge gourd, bottle gourd. They belongs to the cucurbitaceae family. Now last one is our abscisic acid. As mentioned earlier, abscisic acid was discovered for its role in regulating abscission and dormancy. Abscission, the major role is abscission, so it is called abscisic acid. But like other PGRs, this was for its role regulating abscission and dormancy. Other PGRs, it also has other wide ranging of effect on plant growth and development. It acts as general plant growth inhibitor and an inhibitor of plant metabolism. By inhibiting the plant metabolism, it also inhibits the growth of plant, right? AB inhibit the seed germination, stimulate the closure of stomata, increase the tolerance of plant to a various kind of stresses. So it is also called as stress hormone. So it is responsible for tolerance, right, in stress condition. ABA play an important role in seed development, maturation, or what is called dormancy. Seed development, maturation and dormancy. By inducing dormancy, ABA help seed to withstand desiccation and other factors, that is factor means unfavorable for growth. The most situations ABA acts as an, it is, so we can say this one, antagonistic of gibberellic acids, right? So ABA, it is antagonistic to GAs, right? Cytokinins, they are antagonistic to auxin. So we may summarize that for any and every phase of growth, differentiation and development of plant, one or other PGR has some role to play. Such roles could be complementary or antagonistic, right? GA and auxin, they have complementary, but auxin and cytokines, they have antagonistic in nature. Like ABA, GA, they have antagonistic in nature, right? These could be individualistic or synergistic. Individualistic means they acts individually, they can act, or with another hormone, their that is effect is going on. Okay. Similarly, there are a number of events in the life of the plant where more than one PGR interact the effect that event, as for example dormancy in seed or buds, abscession, sensations or apical dominance, etc. Remember, the role of PGR is of only one kind of intrinsic control. PGR, it is only one kind of intrinsic control. Another kind of intrinsic control, it is genomic, right? Now given here, it is given along with genomic control and extrinsic factor. Extrinsic factor means light and temperature, and for light and temperature, it is not in this time, it is not in our NCERT, it is related portion this time, right? Or in old NCERT is given light means what? Phototropism, right? And we set them here we not set them phototropism, it is called photoperiods, right? And for temperature, it is called vernalization. Now this time it is deleted from our syllabus. So many of the extrinsic factors such as temperature and light, temperature means vernalization, light means what? Photoperiodism, control the plant growth and development via or with PGRs. Some of such event could be vernalization, flowering, dormancy, seed, germination, plant movement, etc. It is given. We shall discuss in the briefly the role of light and temperature. Both of them are extrinsic factor on initiation of the flowering. But this time it is deleted from syllabus, so we need not to read this one. Okay. Thanks. If you have any doubt, you can ask in our app. Thanks.